characteristic of the cancer cells. Immortalities, because they increase, they activate the telomerase enzyme. They have the high levels of the telomerase, so they maintain the telomeres or the length. Second, they can promote the growth of the new blood vessels. And third, they have the ability to migrate. We call the metastasis. They can. And here is how the cancer develop. Or we in the hypothetical examples, the cells might first lose activity of a cell cycle inhibitor, and an event that could make cells descendant divide a little more rapidly. Unlike the good or be cancerous, but they might form a benign tumor or mass. So here's in the first step, like the initial mutations here, we have the inactive negative cell cycle regulators. And remember the cell cycle regulator. The negative cell cycle regulator. Remember them? This could be proteins like p53, RB. Remember RB? RB proteins. Also the p53. They call the negative cell cycle regulator. I will put back a little bit to show you what is RB. Remember RB in this step? When I draw here a RB proteins here, and with the a minus here, and also here it could be the CDKs plus cyclines. When we have CDK plus cyclines, I will inactivate the RB here. The RB here no more present here, so we have a replication. So we call them the a negative cell regulator. Here, RB protein of p53 is one kind of negative cell cycle regulator. So when they inhibit the negative cell cycle regulator or regulators, so the next the next generations could rapid or could divide more rapidly, and the next mutations is overactivate the positive cell cycle regulator or regulators. So the positives, this could be the CDKs plus cyclin. Remember in the cell cycles that I remember that are reminding you about the CDK plus cyclin, the positive cell cycle. So in this step, they are divide more rapidly, and in the TOS mutations, they are inactivate a genome stability factor because now DNA is damaged. DNA damage just pass through this the cell division so many times. We have a DNA damage. So if at the end they will affect the genome stabilities, they affect our genomes, and by so many steps, so many cell divisions, we call them the pre-cancerous cell. Okay, so by numbers of cell divisions, the mutation will be accumulate, and in the end we form a cancer cell. So it's not one step that we have a cancer cell, but it's number of steps to prevent the cancer, right? By the first step, when the p53 by the tumor suppressor genes can control our cell division, our cell cycle that's working normal or not, and by the next, by numbers of steps, finally we have the cancer. Actually, it's not easy to get cancer, even the cancer is erased rapidly nowadays, but it's not easy to have the cancer in our bodies. And if they when they divide more rapidly, we have some kinds of tumors, small tumors. We have the benign tumor. benign tumors when they rapidly divide, but when they make the cancer cell, it means they could migrate, and we have the malignant tumor. Okay, so malignant. Benign tumor là các khối u lành tính. Malignant tumor. Malignant tumor là các khối u ác tính. So in this first step, these only benign tumors. But in finals, when we call it that we have a cancer, is mean as is the malignant, acting. So what could be our, I will summarize the step, or when we have the cancers. So first, the mutation happens. A series of mutations eliminates restraints on the cell division. So first we have the mutation, actually the DNA damage, right? If DNA is unfixable, they will go through the cell descendants and accumulate the mutations during the cell divisions is causing the cancer. So first, and in the step number 1 and the step number 2, the cell divides more rapidly. This means we have the tumor. They grow too fast, and tumor cells lose organizations and proliferate quickly. Angiogenesis. What about the angiogenesis? Angiogenesis mean that the cancer tumor can call the blood vessel or surrounding them, sending grow the vessels toward inside the tumors and provide the nutrition and also the oxygen for the tumors. So you know that the tumor is very big, like a big blood is. We have khối u, and angiogenesis is mean that we have the blood vessels surrounding them and descending the signal that's I'm hungry, okay, or I'm hungry, please feed me. And then the blood vessel, you have the blood vessel here, the blood vessel will or goes and feed them. So they can sending the information that the blood vessel send grows the blood vessel inside them to provide nutrition and oxygen. Thus we call the angiogenesis. And the next we have the intravasations, quá trình di căn. Chưa hẳn là di căn. Cái quá trình di chuyển của tế bào ung thư thôi. Intravasation, quá trình xâm lấn. Intervastations in malignant cells spread through the blood vessel and lymphatic system. This is number 3, and number 4 is final stage. We are actually in this case with no curse. We cannot save the human life in this step. It's really difficult for us. So is metastasis. The malignant cell become established in distant organs. So for intravasations, we have numerous of cancer cells go to the blood vessels, but not all of them can survive. It's maybe when they change the environment, we change the pH, we change the or they are stuck at the blood vessel, or they will be engulfed by the macrophage. So macrophages here, or they have been affected by the pH, by the new environments, etc. So number of the factor that could affect the intravasation. So this means that it's difficult for it to have the cancer. We have a number of the process that prevent the cancer inside our bodies, and in intravasations, not all of the cancer cells can go through the intravasation because they have a lot of problems during their process. And also, and by the metastasis, like when the cell reaches other organs, can they grow? Can they grow? at that positions to make the metastasis. That's another problem with the cancers. But if they pass through these 4 steps, we will die. Okay. We have a number of process to prevent them. But if they pass through all the barrier function of our bodies, we will die because of cancer. So what about the mutations? About 10% of human cancer have the hereditary basis, especially for the breast cancer. You remember the breast cancer is or inherit the daughters or the children can inherit the gene that's encoded for the breast cancer from there's father and mothers, and it's into I remember it's 15%. It's 15% of breast cancer because of they received information from their parents' genomes. So about 10% of human cancer have a hereditary basis, and in most cases, the patient inherits some non-function copy of tumor suppressor genes. Here, I remember the RB proteins, the RB genes that induce the RB protein that I showed you. So here, the cancer can induce after the second function or copy of the genes inactive or inactivated by the mutation. So maybe in the somatic or retinal cells, by the mutations, it could be here, this is one copy of mutations. By the another mutation, we have a 2 copy of mutations. That's 2 copy of mutation, 2 copy of mutations, and it's left the cancer cells. Or another case, we have a 2 types of mutation. We have the normal cells. By first, we have just 1 copy of them. And by the second mutation, we have a 2 copy of them. So it's a process, right? 2 copy. And that's why during our life, they it tend to happens with the during the human's age. Like when we get older, it's maybe it have the high chance for it to get the cancer because we contain already contain the mutations, and during the cell divisions is maybe the second mutation happens, so the cancer may happen. And what about tumors? We have 2 types of tumors, right? Malignant is the cancerous form acting, and benign tumors lengthening. So what is the benign tumor? So benign tumors, they do not spread to other parts of the bodies. They're generally localized, and the small size and slow rates of the divisions, and they are closely resemblers and may functions like normal cells. Tự nhiên cô lại quên từ tuyến giáp. Ví dụ như trong ung thư tuyến giáp, ví dụ như các bệnh mà liên quan đến cường giáp thì người ta sẽ phát hiện ra là cái tuyến giáp là có tuyến khối u, và thường u giáp là u lành. Ví dụ như là khi một bệnh nhân mà mắc bệnh u giáp, tuyến giáp là tuyến tiết hormone đúng không? Và khi mà bị mắc bệnh cường giáp thì tức là cái lượng hormone mà tiết ra từ tuyến giáp nó quá nhiều, bởi vì là các u lành sinh ra. So in the benign tumors, they may function like the normal cells. Okay, and they do not break out or originating organs, so they do not migrate. They stay there. They cannot move anywhere, so we don't have di căn, đúng không? No metastasis. And also easy to remove by the surgeries. Those we need to perform surgeries and we remove the benign tumor. That's okay for our bodies. And with some benign tumor that is not grows very fast, maybe the humans can live with them for 30 to 4 years or the whole lives with the benign tumor if they do not grow fast, right? And the second case are malignant tumors. With malignant tumor, we call them the cancer. is actually the cancer because the malignant tumor are acting. So they can invade or spread to other parts of the bodies, and they have the very high rate of the subdivision, and they spread by forming a metastasis zika. So in case of malignant tumor, they have the ability to metastasis. They travel through the circulation system and circulation hệ tuần hoàn nhé. So they travel through our circulation system, and it's really difficult to treat them when metastasis happens. Okay. Here is some frequently certain tissue types become cancerous, and the causes of disease developments and efficacies of treatment are variable. So now think about its type of cancer that's most common. We can see the breast cancer, breast cancer, where the breast cancer. So here we have breast cancer, or maybe or skins is very common, or maybe or digestive system, respiratory system. They're very high level of them, right? Or genital system. Okay, so they are more of the cell in this tissue. They are replacing themselves all the time. So they might have the high chance, or might be very high possibility to have the mutation accumulation, right? Because, for example, the breast tissues is constantly affected by hormones, and is always growing and shrinking, and the lining of the colons is in the digestive system. Columns, they are continuously sloughing off and be replaced. And the same with the skins or cells, and the skins also exposed to the ultraviolet rays. Okay, so all of them divide rapidly and may have a chance to accumulate the mutation. That's why the level of cancer or increase in this kinds of organs. Okay, so so why we don't see much cancer in the heart? Here's ung thư mô mềm. You can see including the heart cancer, ung thư mô mềm nhé. Vậy ung thư tim được xếp vào ung thư mô mềm và cái rate cực kỳ là thấp. Can you give me any hypothesis why we don't have much heart cancer? Why we don't have much heart cancer? So, because other cancers, other cells, they divide rapidly, but the heart cancer, remember that in the cell cycles, I gave you some examples of the cell that jumps, withdraw the cell cycle, and then jumped into the Z0 or Z0, right? And one of them is the heart cell. The heart cells, some of them go to the Z0 state. It means that they do not divide. When they don't divide, no more mutations, and there's no accumulation of mutations. So they have the low chance to have the mutations because they don't replicate. And our cells, the heart is made, or the tissues, the heart is made of connective tissue, and the muscle cells do not turn over very fast, which make them resistant to becoming the cancerous. So connected. connective tissue, Z0. Connective tissue. So that's why we don't have the high level of the heart cancer. Okay, and actually also with the brain. For the brain here, for brains and other nervous system, is most of the case is the as remember the most of the case is in the brain's covering membranes and pituitary gland. So they come from the glial cell, các tế bào thần kinh đệm mà thôi. Các tế bào của hệ đệm thần kinh, tế bào thần kinh đệm. Or others we call the non-neuronal cells. Just because the neuron cells, they are in Z0 state for sure. And whenever we have the heart cancer or the brain cancer, it may come from another path during the metastasis, right? We have the cancer, from example, from the liver, they go to the heart or they go to brains, and that's we have the cancer, the secondary cancer. It's not ours. Less of the case originated from itself. Okay. Khi mà các bạn mà nghe bệnh nhân bị ung thư di căn ấy, khi mà nó chạy vào tim rồi, nó chạy vào xương rồi các thứ ấy, thì đó là cái nguyên nhân mà dẫn đến ung thư tim là do do di căn từ các bệnh có các ung thư khác, đúng không? Còn cái ung thư tim rất là hiếm gặp, bởi vì là thường các tế bào tim nó đã nhảy sang giai đoạn Z0 rồi, nó không divide thì nó rất là ít khi nó bị mutation ở đó. Và ngoài ra thì cái tissue ấy, cái mô cơ tim ấy, nó rất là connective với nhau và rất là khó để phát triển ra cái ung thư. Ngoài ra thì ví dụ như là tế bào não, đúng không? Cô cũng nói là nó sẽ jump, nó sẽ nhảy sang cái tế bào, cái dạng G-nut này, nó không phân chia. Vì vậy mà cái ung thư ở não thì chủ yếu là sẽ tìm được bởi, và nó thường khởi phát từ các tế bào thần kinh đệm hoặc là các tế bào mà mình gọi là non-neuronal cells. So from the cell cycles, from the cell cycle lesson, you will know that the levels of the cancers here are related or equivalent to the time of the cell division. So the organs with containing numbers of the cell which are dividing may have the high risk to get the cancers, right? So what about the cells cycle regulators and cancers? We have two types of the cell cycle regulators may promote the development of cancers. For the positive regulators, is maybe overactivated. We call them the oncogenic. So remember this one of the positive regulator, we have a CDKs plus cyclines also, right? Negative regulators, we call the tumor suppressor, maybe inactivate. You have a P53, we have the RB protein, and maybe P21s, right? So we have a two regulators of the cancer. First, overactivated positive regulator or inactivated negative regulator is make it resulting in the cell or device unstoppable. Okay, for the positive regulators overactive, the growth factor receptors is may send signal even when the growth factor is not there. We call them the overactivated, and the overactivated form of genes we call the oncogenes, and in the normal case they are not yet mutate form, and we call the pro-oncogenes. So from the pro-oncogenes we have a mutation, and these become the oncogenes, and we have cancers. Like here in the normal cells, when they want to grow, when they want to divide, they need to receive the growth factor from the environments, so the cell may divide in proliferations. But if they receive no growth factors, no division. But the oncogene rats here have been mutated into the oncogenes. They will grow even they do not receive any growth factor from the environment. There is mistake here, abnormal here. They receive no information from the external environments, but they say that oh, I received something. Just go and divide. Just go and prorate. So they have mistake, and even we don't tell that cells divide, they still divide because they have a mutations. And the next is the tumor suppressor. Is maybe I show you a lot in the cell cycle lessons about the p53. If we have a DNA damage, first time we stop the cell cycles, take time and take place to repair the DNAs. We call the DNA repairs to repair the DNA damage. But if it's unfixable, we still trigger the hypothesis, and if it worked perfect, no more DNA damage transmit into the next descendants. But if it's not functions, it have been this process inhibit this process. Cancerous may happen. Okay. So now I will show you a video about cancer. But here's. Can I move this?
The human body is a complex and wondrous living machine. Like any machine, our bodies are composed of smaller parts that work together. The various tasks that are performed each day, such as breathing, eating and digesting food, and the movement of our blood, are performed by specialized structures known as organs. Examples of organs include the lungs, stomach, heart, and brain. All of these organs and other body parts like our skin and muscles are made of smaller living units called cells. Our lungs are a good example of how many cells work together to perform a specialized task. The cells in our lungs work together to allow us to take in oxygen from the air and get rid of waste products such as carbon dioxide. Our lungs are composed of many millions of cells working together to accomplish this task. Cancer is the result of a long process that begins when one of the cells in an organ or tissue becomes damaged or altered in a way that causes it to break free from the normal controls that allow our cells to work together in harmony. A group of misbehaving cells can cause the same kinds of problems in a body that a defective part would cause in any other type of machine. A normal cell will divide only when it receives a chemical signal telling it to do so. These signals are interpreted in the nucleus, and the cells reproduce their genetic information and divide into two identical daughter cells through a process called mitosis. Cancer cells do not obey this rule and will divide even if they do not receive appropriate signals. In addition to the signals that normal cells receive telling them to divide, they are also told when to stop dividing. This prevents too many cells from being made. In fact, the cell division process is a highly ordered process. This is a critical issue in cancer because cancer cells do not obey or require normal signals for division. This can lead to the formation of a mass of cells that piles up and may form a tumor. Also different from normal cells is the ability of cancer cells to continue dividing indefinitely. An important point about cells is that no matter what their job is in the body, they all have the same general structure. The cells that make up our lungs, heart, or brain are all similar in their overall appearance, even though their jobs are quite different. Just as the organs that they form are made of smaller structures, cells themselves are composed of smaller parts that help them perform their jobs. These smaller structures are called organelles. Of particular importance in cancer is the organelle known as the nucleus. The nucleus can be thought of as the brains of a cell. It contains the information that acts as the blueprint for each and every one of us, just as a manual would contain instructions to assemble a chair. Specifically, this information is contained within the chromosomes that reside in the nucleus. Individual units of information are called genes. At a chemical level, genes are made of deoxyribonucleic acid, or DNA. All of our cells contain the same set of information. It is only how the information is used that makes them different. For example, cells in the lungs use different bits of the blueprint to do their job than the cells in our stomach. In cancer, changes to the DNA cause some of the genes to fail to perform or to do their job in a way that causes problems for the affected individual. In short, all cancers are thought to result from changes to DNA that alter critical genes and change the behavior of the affected cells. If a change occurs to the nucleotide sequence, it is like having the letters of a word changed. An alteration in a gene is called a mutation. How do all these changes occur? There are actually a number of different things that can cause mutations. Examples include chemicals that can be swallowed or inhaled, such as those found in chewing tobacco and cigarette smoke, and radiation from the sun or artificial sources like a tanning bed. Sometimes mutations occur without any known external cause. They just happen. Certain genes make products that lead cells to reproduce. This would be equivalent to the gasoline system in cars. The genes that are responsible for making cells divide are known as proto-oncogenes. Changes in these normal genes lead to the production of oncogenes. The result may be cells that divide in the absence of proper signals. It is the equivalent of a gas pedal that is stuck in the on position, making a car go even when no foot is pushing down. Genes whose products function as the equivalent of cellular brakes also exist. As a group, these genes are known as tumor suppressors. Humans have 2 copies of each gene, one inherited from each parent. If a single copy of a tumor suppressor is damaged, the other copy is usually able to stop the cell from behaving abnormally. This would be like losing either the front or rear brakes of a car. The car may be damaged, but would still be able to stop. But if the second set of brakes is damaged as well, the car would not be able to stop, just as the cell would not be able to stop dividing if both copies of the tumor suppressor genes are damaged. The process by which tumors cause the body to provide them with nutrients is known as angiogenesis. Like the hungry plant in the Little Shop of Horrors, a growing tumor sends out signals that essentially say, "Feed me." The messages from the tumors cause nearby blood vessels to send over new extensions that deliver food and oxygen. Importantly, the blood vessels also serve as a passageway for the movement of tumor cells to neighboring and distant parts of the body. Spread of tumors to distant locations is of great importance in cancer. About 90% of the deaths due to cancer involve tumors that have spread around the body. The movement of tumor cells to other parts of the body is known as metastasis. Metastasis is a complex process during which cancer cells break off of the original or primary tumor and move through the body to form tumors at new locations. From the point of view of a cancer cell, this is a dangerous and often unsuccessful process. A trip through the body is full of hazards that cause the death of most cells that begin the journey, even tough cancer cells. To begin the process, individual cells must break away from the tumor and invade nearby vessels. The cells crawl along the surface of other cells and the fibrous, stringy structure surrounding them and then force their way in. Shown here is the invasion of the blood supply. Once inside a blood vessel, the cancer cells may perish from a variety of causes. Some cells die simply because they are unable to survive floating around in the bloodstream. Others may become damaged and die when they squeeze through tight spaces or bump into the walls of the blood vessels. Still, other migrating cells may be recognized and destroyed by cells of the immune system. How and where the migrating cells stop is different for different cancer types. Once the tumor cells are no longer moving, they can begin the process of forming a new tumor by leaving the blood vessel and beginning to reproduce in the new location. This does not always occur, and cells that have made it this far may still die or fail to divide. If the new environment is suitable, the newly arrived cell will begin to grow and a new tumor will develop. One way that the development of cancer is prevented is via the death of defective cells. A cell that becomes mutated or damaged will first attempt to repair the damage. If that is not possible, the cells will commit the cellular version of suicide. An orderly process called apoptosis leads to the breakdown of key cell parts and the death of the cell. Cancer cells lose this critical capability and will continue to divide. This can lead to the accumulation of cells that can become more and more abnormal. Due to the high rate at which cancer cells accumulate mutations, a tumor that originally started as a single abnormal cell is actually made up of many slightly different cells. They are all cancer cells and are similar to each other, but they may not all have the same sensitivity to any particular cancer drug or treatment. When this mixed bag of cells is exposed to a drug, most of the cells will probably die. Those cancer cells that are resistant to the treatment may reproduce to reform a new tumor. Importantly, this new tumor will be unlikely to respond to the same treatment. Their mutations make them invulnerable to that drug.
So it's all about the cancer. And I just supplies a small part in the third part in this lesson is about the stem cells. Remember, have you ever heard about the stem cells? I think it's a lot. Wait, tế bào gốc ạ. So what is the stem cells? When we call them the stem cells, it means that the cells have the potential to develop into many different types of cells in the body that we call the stem cells. So they are blank cells, like a paper that you can write anything on it. They unspecialized, that they have no functions, no job, no work. We have a stem cells. and they can renew themselves for a long period of times, and they can differentiate. They have the potential to specialize, to differentiate, to give rise to specialized cells. They are a wise papers. That's when you read unit become a liver cell. It will become the liver cells. And plasticity, this means they can differentiate in numbers of cell type here. From stem cell, one stem cells. They can differentiate as biệt hóa, differentiated into numbers of cell type here. Cells are, we can classify the stem cell by two categories. By the sort of origins, we have embryonic stem cell and also the others. And about the different action potentials, is not all of stem cell can differentiate into different cell types. We have several types of stem cells. Totipotent, tế bào gốc tổng năng, they can differentiate in the full bodies, so the new bodies. Pluripotents, toàn năng, they can differentiate in many different cell types inside the bodies. Multipotents, so đa năng, tế bào gốc đa năng, they can differentiate in some or cell type in the bodies. For example, like the multipotent hematopoietic stem cells may induce several types of the blood cell. Okay, but unipotent is unique. That's tế bào gốc. Or unipotent, this means that it can differentiate. It's just only one cell type. Like, or there stem cells in the skin only differentiates to become a skin cells, not other cell type. So the stem cell will divide very slowly. They can divide to make a new stem cells, or they can make a progenitor. What about the progenitors? What difference between the progenitors and the stem cells? Progenitors is the cell unspecialized that is capable of undergoing cell divisions, and two specialized cells. A stem cells can divide and give rise on additional stem cell and the specialized cells. So progenitors is what the stem cell produce, but the stem cell, they can produce themselves. And here is two case of the stem cells or device. First, in the first case here, we call them the off-ligate asymmetric replication. That is one stem cells divide into another stem cells, and also another daughter cell becomes a specialized cells. Here is we have the hematopoietic stem cells, that the blood making stem cells, or the blood making stem cells. They can divide into another stem cells and another specialized cell like the red blood cells. And the second case of our stem cell divisions, here is the second case. We have the Staurotics differentiation. Staurotics differentiation is in the case here, one stem cells happens differentiate into two daughter cells. This is specialized cell, right? We have the neuron cell and the astrocyte cells from the glial progenitor cells. So they are differentiated into specialized cells, and during that time, another glial stem cell here receive the informations. They noted this, and they make up for the loss of the original stem cell, original stem cells, by undergoing mitosis, and they produce two other stem cells, and we have two other stem cells to maintain the number of stem cells nice and strong. Okay, that's two cases of the stem cell divisions. So, or what could be the source of the stem cells? Are first from the cord blood cells and umbilical cord, or inside our bodies we have the red blood cells, or no, we have the hematopoietic in the mass, in the bone marrow cells. Or we have several cases of the IVS, when the stem cell from the in vitro fertilizations, like we use frozen of embryo states be through the ways, and we can or have the pluripotent stem cells. And also the humans are therapeutic clonings. That's we take the somatic cells biospice, and we put it into unnucleated donor oxides, and they fuse together, and we have the embryonic stem cell. So our ways are also in this case are, I think I need to put a picture, pictures like how we have the from the in vitro stem cells. Like imagine that when the sperm come to meet the eggs, we will have the zygote. Các bạn sẽ có hợp tử đúng không ạ? And we have a zygote, and the zygotes undergo the mitosis, and they will multiply in the four like this. And by subdivisions, they will make a we call the blastocyst, a structure like blastocyst. We have a blastocyst here, and inner we have here we have an inner cell mass. We have one inner cell mass here. Inner cell mass here are embryonic. Or embryonic. And here we have a stem cells. Pluripotence. For the zygote, it's totipotence. Okay. So stem cell can specialize into several other type cells inside their bodies. And also another source of the stem cells is very new, and they are huge discovery. This is, I think that's a huge discovery. You know that in the first time we discovered the stem cells is, or we cannot perform the research by the ethics and science, đạo đức y khoa, because if we want to take the stem cell, we need to take from the zygotes, from the embryonics, and they said that embryonics is the complete human body, so we cannot perform the stem cell research on the embryonics. But for the induced pluripotent stem cell is a huge discovery. So how they can do it? So they can introduce a few specific genes. Here we call the reprogramming factors. They introduce a specific genes into already specialized somatic cell, and then they forgot what type cell they are. When they introduce, they forgot. or they forget, sorry, they forget what type of cell they are. They forget everything after we introduce this specific gene here, and they become the pluripotent stem cells. So we have a high sort of the stem cell to do a research here. And for the next, the purpose of the stem cell, they are we have several various potencies that cell renews. In the normal case, we can use for the organ that maintains or repair, or in the therapeutics, the repairs damaged organs by injecting extra stem cell into into the bodies, and it's always works. It's work for the hematopoietic system, and for the donor Thomas, he received the Nobel Prize in 1990s because his first successful hematopoietic stem cell transplantation in the treatment of leukemia. You know leukemia, bệnh máu trắng. So in the bones, in the bones of the patients, they have the blood cell grows uncontrollably inside the bones marrows here. But now, so we have a number is uncontrollable. But the Thomas know that he can take the hematopoietic stem cell and they inject into the bone marrows and produce normal blood cell for the person again. So the healthy stem cell, the hematopoietic cell here, the hematopoietic system here, it take from the twins of these patients. Or nowadays we can take the healthy stem cell from our bodies. We can take the stem cell, we can cultivate them and then inject inside our bodies. So is more information about the stem cells because it's the additional part. So I don't give you more information. And come back to the cancers. Do you think that for the cancer cells, in the metastasis, they travel all the bodies, and if they can grow in other organs or not is depend on the cell that they are depend on the cell that are they are originated. So you think that if the that's for example the livers or cancer cells, if they are in or differentiated ways, or if they are unipotent, like even they are stem cell, they are unipotent, but when they travel into the kidney, they cannot form the tumor, right? Because the liver cell cannot live in the kidney. It must be at least multipotent or pluripotent stem cells, so they can traverse through our bloodstream and they can reach any organs in our bodies, and they can develop into the malignant tumor. So that the relationship between the stem cells and the cancer, if they are differentiated stem cells, differentiated cells, they could be the benign tumor no more. Okay, if they are stem cells, they might be a malignant tumor because they have ability to survive, to differentiate in other organs inside the body. So it's all about the apoptosis, cancers, and I have a video here, but I will send you later. And also the stem cells. And now it's about the attendance checks. And if you have a question, any question, you can ask me or in the tutorial class also.